research is something which is a little bit boring.
Okay, the first thing what you said, research can be boring. It is not.
Professor Subhash Chandra Lakhotia is a distinguished Indian cytogeneticist and a lifelong distinguished professor of zoology at Banaras Hindu University.
You're doing it for your own choice. You're not doing it because somebody else wants you to do this thing or that thing. I decided not to work in a research institute, which has its own mandate. I wanted always to be a university teacher, when a teacher, and to do research as I like it.
Why Drosophila plays such a significant role and becomes a model organism and opens altogether a different gateway for the researchers? How it is?
Fly is one of the organs which I would always say that it doesn't need a passport or visa to travel to any country. Firstly, it requires very limited laboratory infrastructure. Food is very simple. Where any kitchen can cook it. We don't need anything. Almost 120 years of resources that have been built up and accumulated. See, even the first mutant that Morgan isolated in 1909 is still available. None of the mutants are thrown out. They are maintained and they are catalogued and they are shared. And consequently, the other advantage is that we can make transgenic flies very easily, very conveniently. And the result is now you take any human gene and put it into Drosophila and see where it expresses, what it does, how it does. If we take protein coding genes between Drosophila and humans, 60 to 70% genes are identical. The gene that gives instruction to a cell to become eye is same as in human. Same gene instructs human cells to form an eye, and same gene instructs Drosophila embryo to make an eye with cell. Drosophila eyes are very different from humans.
Yes.
Each Drosophila eye has about 800 lenses. Only one lens.
True.
So the question is, how does it do? And this is where it becomes very convenient to understand what it does to develop. If we compare human and chimpanzee DNA, for example, protein coding genes are almost identical. What differs is non-coding. And that's what regulates. Chimpanzee has a nose, we have a nose, they have two eyes, they have face looks similar, but what is the difference? Regulation. Which way the cells will divide, how many times they divide, when it will divide, these are regulatory events. Why each one of us having same 46 chromosomes, same similar genome, look different, it's this non-coding part that makes this. And Drosophila has stuck with me since then.
Okay. We all know that research is something which is a little bit boring, and that's, I believe, most of the people who doesn't have much exposure into research, they feel that way. Then comes when you say somebody, I work on fruit fly. What is that fruit fly? The other fly which you see in your kitchen. And then, okay, that creature can be so important, almost like a gold mine to a researcher. So to break that myth that it is not just an ordinary fly, why Drosophila plays such a significant role and becomes a model organism and opens altogether a different gateway for the researchers? How it is, sir?
Okay, the first thing what you said, research can be boring. It is not. It is very, very enjoyable, engrossing, and I'm sure even lay people who learn about some research development or something don't think that it comes out of a boring activity. It has to come out of a completely involved activity, and that's what those of us who are involved in research find it that it's engrossing, because you're doing it for your own choice. You're not doing it because somebody else wants you to do this thing or that thing. And so for me, research had been my own choice. And that was one reason long back in my career, I decided not to work in a research institute, which has its own mandate. I wanted always to be a university teacher, when a teacher, and to do research as I like it.
Okay.
Not because because the institute will have a mandate. I may or may not like that.
True.
And then if I have to do it, then the real thing doesn't come out. So I chose to be in a university and to do research as I think at a given time, that this is what interests me, and this is what my curiosity is, and this is what I should do. And that's how it happened. And because of this only, I stumbled into Drosophila. From master's to PhD, I wanted to work with a particular question, but a particular organism. I went to who became my PhD supervisor, to let me work in that. And he said he doesn't have expertise in that, and he can teach me Drosophila, and then I can go back. And Drosophila has stuck with me since then.
Yeah.
This fruit fly is why it has become so important. This was introduced to genetic research in 1909 by Thomas and Morgan in USA. And since then, he developed it so well, and some of the basic foundation that he laid, which is what we now call as a fly room of Columbia University, where he started working, and where these basic foundations were laid, continue to follow till date. Like fly is one of the organs which, till some security issues came about 10, 15 years ago, I would always say that it doesn't need a passport or visa to travel to any country. It has been traveling. Fly, these Drosophila mutants have been exchanged and continue to be exchanged even today across the world without any restriction. No patent issues, IPR issues, and all the mutants that are generated by world over are freely shared, which doesn't happen with many of the model communities. That's one limitation. And these are things that Thomas and Morgan started. Actually, Drosophila started, community started preprints way back in 1933 at Drosophila Information Service. Every year, one volume will come out, printed. But people will send their notes by handwritten or typewritten or something, and will typed out, and one volume will be circulated to all the fly workers, and they were not reviewed.
Oh,
Yes, as I submit, it's published.
so it's more like archiving.
It's not. It's just that was the only source through which we could know what's happening world over. See, try to understand. Before 1990, there was no internet era.
True.
Everything was what comes can go by postal or some service. So how do we get to know who is publishing what? That was our source every year. One volume, entire world, we know who is doing what.
Okay.
And these reports were not peer reviewed. They're typical preprints, and they withstood the test of time. I mean, I've been following them since 1967, I should say, and I haven't come across a report which was later on turned out to be fictitious or wrong or something.
Wow.
And so that has been one thing. The main advantage is that enormous genetic resources that have become available. And every new genetic resource since Morgan's time have been maintained as a full history. Where, when, which day, where it was generated, how it was collected. And these now ultimately have become available in the digital system in Flybase.
Yeah.
So Flybase is, I think, one of the most comprehensive database of everything that want to know about Drosophila species. Melanogaster is, of course, the main species that one uses with, but even other species have become very common now. And all these resources, there are stock centers which maintain these stocks. Like we also maintain all about 2,000 stocks, which we share with anybody who wants that. Like we started about 3 years back a training program for college teachers to introduce Drosophila as a classroom experiment.
Oh, nice.
Because now animals are not allowed to be dissected, so no practice way being done. So we decided that Drosophila can develop as an excellent model. And with this, it's typically people think Drosophila means genetics. That's not really true. Genetics is, of course, main, but we can do physiological experiments, we can do biochemical experiments, we can do neurological, neurobiological, bioinformatic, ecological, environmental studies, all kinds of studies to solve it. Good anatomy, we can do this. And that's what we compile, a laboratory manual written by different Drosophila researchers in the country. We have now compiled about 118 lab practicals, which can be done in a college or university classroom. And this is freely available through Indian Academy of Science, Bangalore. They publish it. Me and Professor Rangnath of earlier of Mysore University, we compiled it, and it has. And based on this, we are now conducting training workshop for college teachers.
Okay.
1 week long workshop where the teachers work like students on the bench from 9 a.m. to 7 p.m. every day, 7 days non-stop. And we have trained more than 100 teachers so far. And what I'm happy that most of them have gone back and set up in their own place where they can teach students to use Drosophila as a classroom. And so Drosophila is not just a small, yes, it's a beautiful fly. We call it Cinderella of genetics. True, true. But it's really not only a beautiful look, but beautiful to work at. And of course, it's polytene chromosomes, the giant chromosomes that way back in 1933, when they understood that they are chromosomes, the paper that described this, the title of the paper was Polytene Chromosomes and the Attack on Gene. Mind, in 1933, nobody knew what gene is.
Correct.
They had only imagined that these are dots on a line, okay, a locus as it is.
Yeah.
But then, when they saw polytene chromosome, T.S. Penter in Morgan's lab, he said that this is the material with which everybody has been dreaming of. We can see genes, we can see how they become active. And polytene chromosome, of course, has been my choice material to work with because of these advantages. And so, now this kind of chromosomes are not available in any other model system.
So that is why, Drosophila becomes a much unique model compared to mine.
Yes, yes. Firstly, it requires very limited laboratory infrastructure.
Okay.
All that it needs is an air condition because it's temperature sensitive.
Yeah.
We can't maintain it beyond 29. 24 degrees, the ideal thing. So in a country like ours, that's one major problem.
Yeah.
So one needs an air conditioner. But other than that, needs most simple things, a stereo binocular and needles and forceps and brushes, and that's it. Nothing else complicated required. But then the kind of things that you can understand from this, of course, as you want to do deeper and deeper, you can have more things, but basic things are very simple. It's food is very simple. Where any kitchen can cook it. We don't need anything to test. Plus, the maintenance is very, and we get large number of progeny in 10 days time, 10, 15 days time. So we can have large number of them. And all these advantages, and of course, the major advantage is almost 120 years of resources that have been built up and accumulated. So even the first mutant that Morgan isolated in 1909 is still available.
Wow.
None of the mutants are thrown out. They are maintained and they are cataloged and they are shared. And consequently, resources keep on increasing exponentially every day, every year as more and more people get involved into this. So these have been main advantages. And the other advantage is that we can make transgenic flies very easily, very conveniently.
Okay.
Because the transposer element that were discovered by Excel in the 1970s or 60s, that has become a Trojan horse. We can put any DNA and it knows how to put it back in the Drosophila genome. And the result is now you take any human gene, can put it in Drosophila and see where it expresses, what it does, how it does. Disease genes, we have models for a huge number of human diseases.
Okay.
Almost all neurodegenerative disorders, they are models in Drosophila. Partly because if we take protein coding genes between Drosophila and humans, 60 to 70% genes are identical.
Okay.
They do same functions.
Yes.
And so one can have a mutant Drosophila where that gene is not function, take a human corresponding gene, put it in Drosophila, and the phenotype is restored.
Yeah.
So like one of the experiments that we showed and which students find it very interesting is that the gene that makes eyes in Drosophila, the basic gene that gives instruction to a cell to become eye is same as in human.
Oh, wow.
Same gene instructs human cells to form an eye and same gene instructs Drosophila embryo to make an eye with cell.
Yeah.
Drosophila eyes are very different from humans.
Yes.
Each Drosophila eye has about 800 lenses. You only one lens.
True.
So the question is, how does it do? And this is where it becomes very convenient to understand what it does to develop. And actually, one of the experiments that we do is, which we teach teachers and we do in classroom is, we have a transgenic fly in which we can take a human eye-forming gene, put it into Drosophila, and let's say activate in what would be a leg. Okay.
Okay.
And you can see an eye in leg. You can see eye on a wing. You can see eye on its back, and so on. This kind of advantage that this can be done very simply.
And sir, it will be a single eye or a compound
It
eye?
will be a compound eye. That's the most interesting.
Okay, so whether the place of the eye is different, but
Yeah.
it's going to be a compound eye.
Because this is how the development program, you see, this becomes a beautiful model to understand how development happens.
Yeah.
You have master gene which gives a command to the cell, okay, you have to form an eye. But what kind of eye it will form depends upon what its next software, so to say, is.
Yeah.
And these are great advantages. Of course, one can do similar thing in other systems, but the advantage that is built into software over decades and decades and decades are really unbelievable.
True.
And that's the beauty of this model.
Yeah, true. I can totally get that feel also. Now, sir, when you're talking of DNAs, I'm pretty sure that when you first, I think in your research you have also mentioned about junk DNA.
Yes.
Right? And this terminology, DNA can also be a junk, because these are two very, very different sort of words put together for the very first time. And how did you come up to this junk DNA
Well,
idea?
I didn't come. This term was given by Susumu Ono, one of the remarkable evolutionary biologists and the human geneticist in US.
Correct.
Of course, prior to that, Crick had given this term selfish DNA,
Yes.
which he called junk DNA. I think the problem which I have come across, which I feel and I've been stating this, is that they were ignorant of biology. Biology doesn't follow reductionist approach of mathematics. In biological system, sum of parts are not always equal to whole, because there's an organization. What they had imagined was that a gene makes a protein, and proteins do the function, bring out the phenotype. So if a gene doesn't make a protein, it is not a function.
Correct.
That was a simple logic. But in the process, what was ignored was that for a function to happen, you need entire regulatory system. When, where, how much that function will be done. Say, as we develop from zygote to become what we are, enormous process of regulation had to happen. So just as I gave the example of eye formation, signal is okay, this cell is instructed you have to become eye, but that's not one step signal that it immediately becomes an eye. It has to follow a process. It has to follow a series of events, and all these require regulation. And so in any genome, regulatory part is much larger than actual information content part.
Okay.
You see, just as we have in English language, if I give an example, we have 26 alphabets.
Yeah.
But then we have different words. You can't put words randomly to make a meaningful sentence.
No.
They have to be in context. Simple example, NO-ON. Two words make very different things. So similar in biological system, which protein will be made when, how much, before another protein or after another protein, it makes a different sense. And this is what is regulated. And I think biologists always were aware of this, that there is a regulation. And actually, 1960s, evolution biologists did state that in evolution, what is important is regulation rather than the protein genes themselves. Our muscle proteins are same, whether it's Drosophila, whether they're roundworm or earthworm or humans. But when and how these are made, that makes a difference. That makes a difference in shape. So my interest, my encounter with junk DNA or selfish DNA happened that I got during my PhD. I got interested in a gene which is uniquely induced with following certain treatment, and this was in 1969. At that time, it was not known that it was known that these what we call polytene chromosome puffs, which are opened up chromatin, that they make RNA that makes protein. But nothing was known that which gene makes which RNA, which makes protein. And because I had accidentally come across an experimental situation where I could activate just one gene and inhibit all others. So I thought I should be able to do this quickly, identify this. Of course, from 1970, I kept on moving from one place to another and couldn't do this simple experiment. Lack of funds, lack of facilities to do all that. Finally, towards late 1970s, we could develop in this lab. Once I moved here to BHU, we fabricated equipment ourselves because we had no money to buy ready-made gel apparatus and gel drying apparatuses. One of my students fabricated with his own hands, but much less cost. And then we found out that this gene doesn't make a protein.
Oh,
And that was the time, this 81. 82 we published this. And 1980 was this term selfish DNA, junk DNA came out. And so we were wondering, did we pick up a gene which is selfish or junk?
right.
I never believed that. As a biologist, having faith in evolution, having faith in power of natural selection, I could never imagine that the cell will make millions of DNA
Which are
copies
no use.
which are of no use because that's the energy currency you are spending meaninglessly. So I thought it must have a rule. Some function it must have. But the selfish gene was what Crick and others suggest was that this gene somehow gets into our system and it's selfish, it just maintains itself without doing anything to the host genome.
Correct.
Susumono junk DNA was that this DNA functioned earlier, but it is just lying unused because that's what the word junk means. There's something which of use in earlier times, but is
Not use anymore.
part of them may be true in certain ways. But then what biological systems do is they repurpose all these sequences. Some old sequence were lying, today it may not have function, but these can be repurposed, and we know a number of examples where this has happened. Repurposing an existing gene, it has mutated, become functionless, and very shortly it acquires a new function. And that's how evolution happens.
Wow.
That's how we get more complexities.
Yeah.
Because see, in Drosophila and humans, if they share 70% of the protein coding genes, why should we be so different?
True. Absolutely.
Many of these have been repurposed in different ways. They are regulated in different ways. So that's what my belief was, and I kept on working on this, and I'm glad that we kept on. Interestingly, my research in India for this gene was funded, not very much, but okay, it was funded. My competitors in US and Germany couldn't get fund for selfish DNA. They had to stop it. So there was a time for almost 10 years or 20 years, I was the only one in the world working on this gene. No one else was working because nobody would get funded. But now we know non-coding DNA and RNA are very, very important.
Yeah.
And this is something that I always believed. In 1996, I had actually predicted that non-coding RNA is going to be very important. And that's what has happened now. Like if you take in human genome, we say protein coding genes are less than 2%. 98% was selfish. But it's this 98% that makes us human. See, if we compare human and chimpanzee DNA, for example, protein coding genes are almost identical. What differs is non-coding.
Non-coding.
And that's what regulates. Chimpanzee has a nose, we have a nose, they have 2 eyes, they have face looks similar. But what is the difference? Regulation.
Yeah.
Which way the cells will divide, how many times they divide, when it will divide, these are regulatory events. Why each one of us having same 46 chromosomes, same similar genome, look different, is this non-coding part that makes this.
Wow.
And that's what we now learn. And so I'm glad that I didn't give it up, because we did discover new structures in Drosophila for this. I mean, one of what we now people talk about phase separated entities, we discovered one such entity and we gave a new name for this as omega speckles.
Yes.
In 2000.
I thought of touching that
and,
also.
and that was something very interesting, because when we saw it first, it was my student Prashant, who is now in Illinois, University of Chicago, Illinois.
Yeah.
I'm sorry, University of Illinois. He saw it and he couldn't believe that this is something that we are seeing. But that was a remarkable thing to happen.
And now, of course, we know this gene is much more complicated. It makes almost 7 transcripts, which can do very, very functions. And I continue to be engaged with this gene for now how many years? Let's say 1970, or even if I take 1980 to 45 years.
Yeah, it's around 40, 45 years.
45 years. And I think if I have one more life, I will not be able to solve the riddle of it.
But the important thing is, if you have one more life, you still want to work on this.
Yes.
That's
Because it's so interesting and so intriguing.
yeah. So that is what the spark said. I want to see what is amazing about it. People might say 40, 45 years is a very long time. What is going to happen? But what keeps you ignited, ticking a
Well, you see, it's like this. You know today something new about it. Now you ask next level of question. Because only when you know this,
yes,
you know what you still do not know.
yeah,
And that's what
keeps you moving step by step.
it keeps moving step by step. But one thing more I will say.
Mm-hmm.
So your beginning question that people think of researchers very queer persons.
Yeah.
Okay. It's not really so.
Yeah.
I'm an ordinary person. Interested in this. I do all kinds of other activities. I have my own hobbies, my own work at home. I mean, I can do all kinds of work at home as well. But it's just the question of remaining busy, remaining happy. That's what is more important.
Yeah.